So today we are taking a look at what is called the central dogma of science. So a dogma is like this foundational belief, and everything else we know is built around a dogma. So for instance, our understanding of the solar system and of the planets and of space, really, a lot of it has to do with the planets revolving around the sun. So in the solar system, the dogma is, the central belief is that the sun's in the middle and everything revolves around it. If that's not true, all of our other understandings and beliefs really fall apart. So that would be for like space. What we're covering today is the central dogma in biology. If this falls apart, if this isn't true, our entire understanding of biology is, well, tough to say. All right, before we get there though, quick notes here on RNA. So it's DNA's little brother. RNA is very similar to DNA. Very similar. So similar, we're not even going to draw it. Pretty much the exact same. Collective, yay, goes up. Yay, I was happy. Not diagramming RNA. There are some differences, so we'll make sure we make note of these. First of all, the name, RNA. It's different, right? An R instead of a D. So just like DNA had the 5-carbon sugar, remember that pentagon that we always drew in the middle? In DNA, that pentagon was called deoxyribose. In RNA, it's called ribose. Again, that's kind of the difference between DNA and RNA. Right there in the names. Another big difference. Again, so far, no difference. DNA had A, G, C, and T. RNA has A, G, C, and then U, uracil. And that's in place of T. It's otherwise identical to T, thymine. So they're both pyrimidines. That means they're the shorter one with just one ring. Uracil, just like thymine, makes 2 hydrogen bonds instead of 3, and he bonds with A. So he's really very much identical to T, but he's found in RNA, whereas T is found in DNA. Ooh, what's going on with me and letters? Yikes. RNA is usually single-stranded. So when we talked about DNA, we always talked about their anti-parallelism, the 2 strands that base pair in the middle? Well, RNA just has one strand, not a double helix. And another difference, there are a lot of different types of RNA. DNA is just DNA. We say DNA, we're just talking about the double helix, it's DNA. Its sole job is to act as the genetic code for life, and that's pretty much it. RNA has a lot of different types that do a lot of different things. It's almost more protein than DNA. It fits into proteins, it does different jobs, it has specific tasks. So RNA, a lot more various than DNA. So we have, I think there's an A in there. What do you guys think, A, messenger or messenger? I'll keep E. We have messenger RNA, which we'll just call mRNA. Can use a little M there for messenger, shortens it. Transfer RNA, which we just use a little T for that one. Ribosomal RNA, which we use a little R for that, rRNA, like a pirate R. And there's actually more too. It's like snRNA, small nuclear RNA. There's another one I can't remember the name of. There are a lot of different RNAs. We'll concentrate on just these three for this class stuff. But again, RNA, almost more like a protein in a sense. Because it does a lot of different things as opposed to DNA, which just contains the genetic code. Questions so far? Doing well so far? Cool. Now let's talk about transcription. And what's diagram, huh? What would a day with Richardson notes be like without a diagram? Well, hey, there's our new favorite shape, huh? If I could have a superhuman power, I would like to be able to draw a DNA strand better. That'd be my superhuman power. We all get one. All right, so this guy so far should look familiar to us. Here is the new part. This is mRNA. Just like DNA, it gets copied in the 5' to 3' direction. So you're always going down, 5' to 3'. And he's being built, he's being made as a copy. What we're doing is we're opening up the DNA, we see that here. We're copying the RNA, and then it kind of peels off, and now it's just a single strand. At the end, the DNA closes back up. It re-anneals and goes back to its double helix. We're just opening it up very briefly, copying our strand of RNA, mRNA, and it leaves and peels off and goes and does its thing, and the DNA copies back up. The strand we're actually using to copy is called the template strand, the strand of DNA, versus the other strand that we're not actually copying is called the coding strand. Seems backwards, right? Well, if you think about it, we're using this bottom strand here to make complementary bases with the mRNA. We're matching it up. So if this were an A right there, this one would be a U. If this were a C, that would be a G. So the mRNA is actually not going to look like the bottom strand. That's just the template strand. This mRNA is actually going to look like the top strand. So that's why the top strand is called the coding strand. That's the one that's actually going to end up going on to become the gene and is going to get turned into protein. The top strand, not the bottom strand. So what we have happening here, this process is called transcription. And we are transcribing. So we're opening up the DNA, we see that there. And then we're copying it or transcribing it into an mRNA strand. Good so far? All right, next up, let's talk about what you guys did, your LT3, what your e-learning was last week. mRNA editing. Nice, thank you. I almost adjusted my monitor as if that would do anything. So here's our mRNA strand. Again, we transcribed it from DNA. And the next thing that's going to happen to it, and this is before it even leaves the nucleus. These spliceosomes, these are little proteins. These spliceosomes kind of make these loops. They push parts of the DNA back. These parts are called introns, and they're going to get edited out. They're going to get cut out. The parts that we do want, when we make these loops, the parts that we do want, we kind of bring together, and they're able to splice these proteins, splice these ends back together, and that cuts out the loops and leaves you with your desired message. Again, that's hopefully what you guys did for LT3. The region between X's, so this is like double X, that region in between, you guys cut out. You deleted it. You drew a line through it. It's gone. Same thing's happening with our mRNA. Spliceosomes begin pushing it back and then cutting it out and linking the two sides together. This mRNA editing process, what you guys did for your LT3, we're cutting out what we call the introns, and we're splicing back together the exons. The introns remain in the nucleus. They should probably get recycled back to a new strand, whereas the exons can now leave the nucleus, exit the nucleus, and get translated. So I'm going to flip my page for the next note. Anybody still need this? Everybody here in the classroom has their pencil down, so people at home, last chance, speak up. Still need to see something here. Cool. All right, so sorry, this is kind of a continuation of what we were just doing. We just didn't have enough room on that other page. All right, real quick, I'm going to go back. Someone has to go back. All right, the last couple of moments. All right, so I'll leave it here for just a minute. Else can shake out their hands, get some blood flowing again. How's your day? Been good so far? Better being here than being at home?
Yeah.
Really?
Yeah.
Even though you guys don't say a word?
The information is kind of like processed.
Really? So from an educational standpoint, it is better to be here. Hmm. You know, I got a good way we could test that. Tests. You guys don't care that much, huh? All right, I'm flipping back. We can come back to this later. Man, a little bit of first semester coming back to us. So our next stop or next step here is translation. And in translation, our mRNA is going to leave the nucleus. So we have our mature mRNA. We have edited out the pieces we don't want. And it's going to travel to a ribosome. We talked about ribosomes briefly last semester. Remember, those ribosomes can either be free-floating, that means just kind of on their own, just anywhere in the cell, or attached to the rough endoplasmic reticulum. That's why it's called rough, because it's got these bumpy ribosomes on it. So either or, this mature mRNA strand is going to travel to a ribosome where it can now be translated. Quick note then on the ribosome. It does almost kind of look like a hamburger bun. At least that's what I think it looks like. It's got two subunits, a big unit on top and a small unit on bottom. And those two units kind of come together on the RNA. So if there's a strand of RNA, it's like your bottom strand latches on, and then the top strand latches on. So they do kind of work in unison. All right, so here is our ribosome, and it's really not much. I mean, in the sense that last semester we called it an organelle. And I think a lot of people wouldn't call it an organelle. Number one, we always say that bacteria cells, prokaryotes, don't have organelles. But they do have ribosomes. Every cell on Earth has ribosomes. You have to. How do you make proteins without ribosomes? I don't know. I don't think you do. So everything has ribosomes, including eukaryotes that don't have organelles. Also, all of our other organelles are membrane bound. You guys remember the phospholipid bilayer? That surrounds every organelle except the ribosome. The ribosome is really just a collection of a couple of proteins and some rRNA that almost acts like rope to hold them together. So it's nothing sophisticated. It's really just like a large, complex protein, hardly an organelle at all. But it has its job, and it's a very important job at that. And it latches on to the mRNA strand. Like the bottom strand grabs a hold of it, and then the top strand comes in on top. And it kind of sandwiches the mRNA strand. That's how it's going to read it. Before we get to the reading of it, one more quick note over here on a new player. This is tRNA. Sorry about that. Okay. Let's have you move under the quotes flag, and there's a plug right behind that. So when you say you need to charge it, are you under 20%, or are you like at 11% and it's about to turn off? Yep, get yourself plugged in. Everybody else doing all right with battery juice? All right, almost there, guys. Like one more sentence. I don't know, he was checking your watch, like, wait a minute. We still got a lot of class left. What are we gonna do? When's the last time we had like 40 minutes left and we were done with notes? All right, so here's what's going on. Here's tRNA. This section right there, that's what we call the anticodon. It'll make more sense in a second. And then up here, if you didn't know what the AA stood for, that's what's called the amino acid. Okay, so what we have happening here So what we have happening here what we have happen is the ribosome reads the mRNA strand, and he does it 3 bases at a time, 3 letters at a time. Each letter, each 3 letters, is a word, and in the words of the genetic language, the words are called codons. So 3 letters at a time, each codon at a time, tells the ribosome to call over a different amino acid. And each amino acid, which is, with its anticodon, and that's why it's called an anticodon. It's an opposite of the codon. The mRNA strand has a codon. The tRNA has an anticodon. They match up. Any 3 letters you want, but no T's. So A, U, C. Well, he calls over UAG. And attached to UAG is one particular amino acid. Questions? Fantastic. Who's ready to learn a new language? I'm going to teach it to you in 10 minutes, whole language. Oh, yeah. For those of you at home, go to Canvas. Those of you here, don't worry, I can get you something. Although it is on Canvas when you need it also. Those of you at home, go to Canvas, go to Modules. And for today, you guys will see something called the codon wheel. I got one for you guys. And you guys will need to open it up, and the codon wheel looks something like this right here. So again, if you're at home, open that up on Canvas. Go find it, open it up. I'm going to hand these out to people who are very young. So this is what we call the codon wheel, really cool instrument. And it allows us to read the genetic code, 3 letters at a time. You start in the middle. Again, you got one of 4 options, right? If you're making a 3-letter code, you got one of 4 options. You can start with A, G, U, or C. And then you work out. Let's say you start with A, well, then you've got 4 more options. After A, you could go G, A, C, or U. And then once you get the first 2 letters, you have another 4 options. And depending on whatever three-letter code you have, there is your amino acid. You'll notice all 20 of the amino acids are listed around. These are the amino acids. And you put those amino acids together and you build a protein. A couple things I want to point out. I don't know if you guys can edit this at home, make a note somewhere or something. This one right here, very important. A, U, G. I always like to think of it like August, the month of August. A-U-G. This is what we call the start codon. Guys, you can't start unless you have a start codon. The ribosome doesn't just randomly start going. He actually has to read the start codon, which is always A-U-G. Then you'll notice there are 3 stop codons. When you read a stop codon, guess what? You stop, you're done, your protein is through, it's finished. Those 3 stop codons are U, A, U, A, G, and U, G, A. Those all result in stop. Questions so far? Cool, got a little bit of practice for you. Well, again, you guys got some homework on doing this tonight, so to make sure you guys all see all the tricks and all that. So real quick little practice. So how this usually works is you are given a DNA code. And that'll look something like this. See? There could be anything here either. So you're given a DNA code like this right here. And what you normally need to do then is transcribe it first into mRNA. So we match up our letters. C matches up with G, right? T matches up with A. Watch out for A. There is no T in RNA, so that's a U right there. C becomes G, C becomes G, C, G. T matches with A. C with G, C with G. A matches with U. Watch out for U. G, A, A. Now watch out. As we get started next, I can tell some of you guys working on your homework already. Stop. Stop. Watch out for this because it'll get you, and then you're all messed up and you get lots of wrong points because you just aren't paying attention. AA stands for amino acid. This is going to be our sequence of proteins. Guys, this is big. This is key. This is important. You can't start until you read the start codon. People mess this up all the time. My first 3-letter sequence, GAU, is not the start codon. The start codon is A, U, G. That's my start codon. I can't start until I read that. So it's not GAU. Okay, let's move down one. Ah, there it is. AUG. There's my start codon. What amino acid is it? Well, honestly, it's methionine. We're not going to write that whole word. You can if you want to, I suppose. But I just write the first 3 letters. So I'm going to write MET for that one. MET. Hey, after that, once I get my start codon, this is a trick. You don't have to do it, but it's going to make your life a whole heck of a lot easier. Break everything into 3s. Again, we're reading 3 letters, 3 words at a time. 3 letters is 1 word. So it just helps kind of the eyeball to see where you're at if you break it into thirds, or 3s, I should say, like that. All right, very good. Our next codon is GCG. Again, using the codon wheel, we start at G, second letter C, third letter G. Hey, alanine, that's my favorite amino acid. How about them apples? Then we go AGG, A, G, G. Arginine, ARG. Next up, UGA. G. Hey, that's a stop codon. Hey, guys, you're done. Do not go past that. Oftentimes on a test, I give people this big long line and they have to do this huge translation, or so they think. But really early on is a stop codon. You guys have no idea, makes my head hurt. How often people write the word stop and just keep going. Or they write the first three letters, STO, and then they keep going. I'm like, no, no, no. Stop means stop. If you hit a stop codon, you're done. Your protein is over. That's it. Calvin, there you go. You just learned a whole new language, the genetic language, and it took less than 10 minutes. How about them apples? Questions?
Could you go back to the wheel again? Like the wheel
What?
that has this, the start and stop thing?
And again, this is on Canvas. You guys can find it on Canvas. Just make sure you notice, add, take a note if you need to. AUG is start. 2 stops. 3 stops, technically, because there's 2 of them right there. One second. Basically, yes, but yeah, 1 second. Okay, so 9.3, this is posted. It's due by Thursday. If you haven't done LT3, guys, get that done. It's due tonight. But here is your homework for tonight, due Thursday night. Yeah, look, a little bit of what we just did. So that's pretty cool. Huh? Pretty cool. Questions? All right, well, how about the apples? We got half an hour till lunch. You know, last time we had half an hour free time in this class. Yeah, I don't know. People at home, have a good afternoon. I'll see you guys Thursday. Unless you want to stick around and ask questions, feel free to do so. But I'll see you Thursday. Yeah, you guys can. Thanks for sticking in there. I just, Ava, I just had you stay on for an extra second or so because last period I let people in class leave before I showed the homework, and then everyone on Zoom thought they could leave too, and so everyone left. I was like, hey, so thank you for sticking around. You guys did great. You're a great audience. What are we gonna do for the next half hour?
Hey, how am I supposed to do question 1 on 9.2? Like, can you help me get those done?
On 9.2?
The quiz.
Okay, what, Payne, why are we always so far behind on doing stuff?
Not